Would you be able to say how diverse the bugs are on this plant? Where would you start, and how would you do it so that you could realistically and accurately compare your results to what someone else could come up with? I'm going to talk about quantifying biodiversity and where current best practices come from, information theory. First, I'll talk a little about what biodiversity means in a scientific context. Then, together, we'll work through an example of quantifying the biodiversity of bugs on a plant. Then I'll introduce information theory and we'll calculate a single number, an index, to represent the amount of diversity in our sample. We'll then transform that index into something a little more meaningful to biologists, effective number of species. And I'll wrap up the video with some examples of why this type of work is important and how it's applied. Jump between the different chapters using the navigation bar or timestamps in the video description. Biodiversity is the amount of variety of life. Okay, that's kind of a lot, so for it to make sense or even be remotely usable, we need to consider biodiversity at different scales. Think about this lot. What's the biodiversity of a city lot? What are all the different plants, animals, fungi, and microorganisms that live in a lot? How can I generalize this lot to other city lots? How many of each of the different types of organisms are there? And how much variety is there from, say, this one mallow to the one right next to it? Each of these questions is looking at a different scale or focus of the broader question, what's the biodiversity of a city lot? Imagine a tiny lot with just 5 burdock plants and 5 dandelions. Is that more or less diverse than a lot with 5 burdock plants, 4 dandelions, and a clover? So there are different measures of biodiversity that depend on how we weigh the importance of rare species. The simplest way of measuring diversity is just counting the number of different types, usually species, and this is known as richness. Moving beyond that is a summary of multiple different factors. Most often in the case of a lot, say even just the plants, an extra obvious factor on top of richness is the evenness, or how spread out the count of those species are. As in, a lot with 5 burdock plants and 5 dandelions is more even than one with 9 burdock plants and 1 dandelion. Ecologists have developed methods of summarizing the evenness of species in a location as a single number, which lets them compare evenness across systems or scales over time. These summaries are called indices. And often multiple indices are used, but for this video we'll focus on the most used index, the Shannon Diversity Index. Let's work out an index together, and let's work from an interesting and manageable scale like the diversity of bug types on this one burdock plant. So I'll show the different types I roughly identified and give a count of how many of each there are on this plant. The numbers aren't exact and were chosen to help prove my point, but are mostly realistic relative to the actual proportions. I'm also only including the 4 most abundant types. By far the most abundant bugs here are aphids. These ones are maybe Aphis fabae, probably at least genus Aphis, and on this plant they're being farmed by ants. Here's what they look like standing up straight with intact wings, but all of these either have no wings or have had their wings clipped by the ants. Their heads are jammed in the stem drinking the sap, and those are their back legs flung up in the air. Let's say there were 400 total aphids on this burdock. The second most abundant bug were parasitoid wasps. What you're looking at is actually the husk of a dead or dying aphid. A wasp will lay an egg inside a living aphid, the egg will hatch, and the young wasp will consume the aphid from the inside out, emerging from a hole like this one. I didn't actually see one of the wasps outside of an aphid, so I can't really get specific about their identity, but let's say 200 total wasps living inside aphid husks. Next are the ants. There's one type of ant species present, they look to me like carpenter ants. They're farming the aphids, so they milk the aphids by massaging them with their antennae, then eat the honeydew that the aphids secrete. In return, sometimes they protect the aphids from nastier predators like ladybugs, but also usually clip their wings to prevent them from moving around too much, and sometimes just eat them anyway. Let's say there were 100 total ants. And lastly, the lady beetles. There were a few adults getting in scuffles with ants, and lots of larvae of pupae. Probably all Asian lady beetles, definitely Coccinellidae. Including all the life stages together, there were about 100 lady beetles. Okay, so to recap, our plant had about 400 aphids, 200 parasitoid wasps, 100 ants, and 100 lady beetles. The origins of Shannon diversity come from a 1948 paper by mathematician Claude Shannon, describing information theory. He was trying to come up with a value that described how much information was contained within an average message. This measure of information could tell us how much space it would take to store or compress a message, and how many messages could be transmitted. We call the measure Shannon entropy. I won't get into too much detail about information theory, If you want a deeper dive, check out this video connecting Wordle to information theory by 3Blue1Brown. So not too long after Shannon's paper was published, information theory was adapted to describe information within biological systems, Shannon diversity. Let's see how we can apply Shannon diversity to our little system and come up with a single number, Shannon diversity index, to summarize our bug diversity. So if we want to represent diversity, we'll need each type of bug and their relative proportion of our total to contribute something. This is where richness and evenness come in. For each bug, we need a way of representing its contribution to both evenness and richness. Then we need to sum those contributions of all the bugs we're counting. The only real hard piece of numerical data we have for each of the bug types is the count. Consider each count within the context of the total number of bugs and it becomes a proportion. So add up the counts of each bug, 400 plus 200 plus 100 plus 100, and our total is 800. Then divide each individual by the total, 800, to get each proportion. Then simplifying the fractions, we get these for our proportions. Okay, I'll insert our proportions into our equation because we'll definitely use those, but we also need a way to scale these proportions to reflect the fact that having more bugs from rare species contributes more to diversity than bugs from a common species. In other words, we want bugs with lower proportions to have more of an effect. We want to scale them up an appropriate amount. This scaling is definitely the weirdest piece of the puzzle. How much to scale by and what's the reasoning? Try thinking of it like this. Imagine randomly picking a bug from our burdock plant and thinking of the scaling term as a measure of surprise in the identity of the bug. How surprised are you to see an aphid? Unsurprised. Ant? A little bit. I would have bet I'd pick an aphid. I bet I'd be X times as likely to pick an aphid as an ant. And that's it. That's the number we're after. How do we represent something like X times as likely to pick in a number that we can insert into our equation? One that works for all proportional differences, no matter how high or low the richness or evenness are. So Shannon's implementation of this used logarithms. Log base 2 is the easiest to get intuitively, so let's use that. Okay, and now we need a number to include in our log. We can't just use a static number, a constant, because we want the term to vary between bug types. Remember, aphids less surprised, ants more surprised. So we need to use a variable. What if we repurpose the already defined variable that's the proportion of each bug? Using this variable in the log, we can make it so that as the proportion gets smaller, that number itself contributes more and more to make the total larger. This would increase the contribution of rare species, but in a reasonable, scalable way. So if we just plug the proportion in, there's a little bit of intuitive weirdness, so bear with me. Let's start by taking the log of 1 over the proportion. So for aphids, 1 over a half simplifies to 2. Let me plug this in as a scaling term. Now while you can just do the logs using a calculator, I want to break it down so it makes sense. I'll explain using the aphids log base 2, then work it out for the others. So, by definition, log 2 is actually just another way of saying 2 to the power of what is equal to 2. And in words, for example, it's the same as saying, given all the bugs, how many times do you have to split the total in half to equal the number of aphids? Once. The aphid scaling term, log 2, is equal to 1. Okay, look at wasps. 1 over their proportion, which you can see at the top is a quarter, so 1 over a quarter is 4. Now log 4 is the same as saying 2 to the power of what is equal to 4. Given all the bugs, how many times do you have to split it in half to equal the number of wasps? 2, twice as surprised. And ants and ladybugs? 2 to the power of what equals 8. 3 times the level of surprise choosing an ant as an aphid. Split the whole collection up 3 times before you have your number of ants. Or, I bet I'd be 3 times as likely to pick an aphid as an ant. As an aside, usually with Shannon diversity, for the scaling factor you'd use ln instead, which is log base e. But you could use log base 10 or any other base, and after the next section you'll see that it doesn't matter what base you use. And another aside, remember the intuitive weirdness I mentioned and how we use 1 over the proportion? So you often see the equation as taking the log of the proportion directly without the 1 over, but in that case it's the negative log. These are mathematically equivalent, here it is written out, or just take my word for it. I use the interpretation I did because I find it more intuitive. Okay, so we have these surprise values, the log term we just worked out. Each multiplied by the proportion, then summed. But why multiply? Well, it's averaging the surprise out. This way, we're measuring how surprised we would be on average when we randomly sample an insect. So you plug this whole thing into a calculator and get exactly 1.75. So this 1.75 is what Shannon would have called entropy, or measure of average surprise. 1.75 is a measure of how surprised we are, on average, with any specific result when we randomly sample bugs from this plant. Okay, now to go from entropy, or this index, to an effective measure of diversity, we need to go one more step. For motivation, imagine this example. A community with 8 different bug types in perfectly equal proportions, say exactly 100 of each bug. So here's that formula from before. Now the entropy H prime is this added up for each different bug. Plug it in, you get 3. Think about how they compare. A community of 8 different bugs, all equally common, none of which is rare, is more diverse than this difference reflects. It should be at least twice as diverse. More than 1.75 to 3. Okay, here, look at an extreme example. Imagine 50 bugs in equal proportions. If we want our measure to be robust, it needs to show that this community is way more diverse. The entropy is 5.6, which is a little less than twice as much as the entropy for our 8-bug community. But we need the difference to be more representative. Now to get there, imagine a hypothetical system with the same index as ours, 1.75, but with a different composition of bug types and proportions. This time, assume a fully equal spread or evenness of types. We know it can't be 8 different equally abundant bug types, because that gives us an entropy of 3. So how many bug types would it have? Let me say that again. We want the same index, 1.75, as our original mix of bugs, but with an equal spread, so not weighted towards aphids like ours. How many different bug types would achieve that? We go back to our equation and start plugging in random values, trial and error, until we get something close. But there's some math we can do to just give us a direct, exact answer. The derivation's beyond the scope of this video, sadly. It's a little more involved than just solving for the number of species, but in the end, the solution is to exponentiate the entropy, or the H prime. So we take our original log base, which was 2, and calculate 2 to the power of H prime. Without rounding the entropy, we get about 3.4 for our original mix of bugs. 8 for the 8 equally proportioned mix, which is great, it makes sense for it to be a little over twice as diverse. And for the group of 50 equally proportioned bugs, we get 50, which is, way more diverse. The description and units of this newly transformed number supports this. It's known as the effective number of species, or types in our case, which is a solid, easily and effectively comparable measure of diversity. So to summarize the jargon, H prime gave us entropy, or the Shannon index, and now exponentiating it gives us a true measure of diversity, or effective number of types. Through this transformation, our collection of bugs is easier to interpret on its own. Sure, there are 4 bug types, but because there's so many aphids and relatively fewer of the others, it's kinda like there are 3.4 bug types. Ultimately, it can now be compared to other systems of any size that have been measured and transformed in the same way. So one last little math note. Remember how we used log base 2? I said people often use ln, or log base e. Well, if you do it like that, at this step you would do the same thing here, but with e instead of 2. So whatever logarithm base you use, that's the base you use when you exponentiate to calculate the effective number of species. Biodiversity is obviously really complicated, and different degrees of biodiversity, high or low, have all sorts of effects. Measuring it or estimating it lets us better understand those effects. Biodiversity is dropping, and it's going to keep dropping as human populations and their activities keep expanding. So one important use of biodiversity indices is in determining thresholds for the loss of ecosystem function. Turns out the impact of biodiversity on an ecosystem changes more dramatically with larger losses. Small losses in biodiversity don't have much of an effect, but bigger losses start to compound changes in function. Here's a quote from this 2012 paper. We do not yet have quantitative estimates of the level of biodiversity at which change in ecosystem functions becomes significant for different processes or ecosystems. Well, if we're going to come up with these estimates, we need to use indices. Different levels of biodiversity can also change ecosystem services. When land's used to grow food, for example, that land's structure, composition, and functioning is dramatically simplified. This is great for, say, the yield of whatever crop's being grown, but not so good for some other ecosystem services like climate regulation or disease control. Simplified landscapes also have much lower biodiversity, but we're still not clear about this relationship of lower biodiversity and reduction in some ecosystem services. We have some pieces of the puzzle and enough to draw some conclusions, but more research is needed to complete the puzzle, which means the use of indices. Finally, when things go wrong, more biodiverse ecosystems see less loss in function and service and bounce back faster than less biodiverse ecosystems. That degree of loss and rate of bounce back are measurable, so we can use quantification of biodiversity to estimate levels of resilience. I laid out the Shannon Diversity Index here, but there are other indices. They each have their own strengths and weaknesses, and rather than talking about the pros and cons of Shannon diversity or any other index specifically, I wanted to close out the video going over the range and limitations of biodiversity studies in general. An index is only as good as the data. Often data has problematic sampling bias. Easier to sample species are counted more often than difficult to sample. More species in larger numbers are found in areas where people search more. Small and large changes in timing, geographic, or environmental variation that affect where we find different organisms aren't always covered. A single number, a diversity index, can say a lot about a collection of different life forms, but how that number is derived can be incredibly sensitive. And lastly, one of the things I love the most about this is the scale or scope of measuring diversity. You can perform your own estimates with things like bugs in your compost or plants in your yard. You can get as complicated as you want down to species or variety level, like classifying each species of aphid, or just go broadly by type like I've done. Scientists use a huge range of strategies depending on the question and scale they want to address. Some studies look at the biodiversity of microbes in our guts by sequencing DNA and building an index based on the tiniest differences in genes, while some studies build indices on broader or just alternate categories of diversity, like different plant families, or the diversity of functions organisms provide to an ecosystem. So what's the biodiversity like in your backyard, or your favorite park? That's all I have for you. If you made it this far, thanks for watching. Most of the footage was shot on the Concordia University Loyola campus. Thanks to Dr. Eric Pedersen and the Concordia Experiential Learning program for making this video possible.